So this unit is about the cell cycle, which is basically the stages that the cell goes through during the course of its lifetime. Some cells will go through these stages thousands of times. Some cells will never really get through it one time because it'll kind of stop or arrest and not actually finish going through the M phase or the mitosis phase. So we have what's called interphase, and then we have mitosis. And interphase is kind of like the resting part of the cell. It's kind of getting ready for this cell division or cell reproduction to occur. So you have G1 stage where you have a lot of growth occurring. You have organelles copying themselves. You have the S phase. S stands for synthesis. And this is where DNA is going to make a copy of itself. We'll talk about that process a little bit later when we get to genetics. And then you have the G2 phase where all of the cell is getting everything else ready in order to divide into two new identical cells. The end result of the cell cycle is two identical cells. And this is important because like when our skin cells die and fall off, we want to replace them with other skin cells. So this is the process the cell goes through in order to do that. Mitosis then is the reproductive stage of the cell cycle, and it consists of five phases: prophase, metaphase, anaphase, telophase, and then cytokinesis. The cell spends the vast majority of its time in interphase. All cells do, except cancer cells are going to spend more time in mitosis, more time actively dividing. Cancer is a disease of the cell cycle. Here's another diagram that's kind of showing you the stages of the cell cycle, including each individual stage of mitosis. So G1, S, G2, growth stage, synthesis, preparation, and those three are interphase, and then prophase, metaphase, anaphase, telophase, and cytokinesis. Again, the end result is two identical daughter cells. I want to briefly go over with you the stages of mitosis because they're important. So during prophase, which is the first stage of mitosis, the chromosomes basically squish themselves together, they condense, and so they actually become visible under the microscope. You can see them. The nucleolus disappears, the nuclear membrane disappears, so everything is just floating around in the cytoplasm. There's really no nucleus at this point. And you have these fibers that start to form that are called spindle fibers. So basically everything's floating around in the cytoplasm. There's no nucleus. The next stage is metaphase, M. Think for M for middle. The chromosomes are going to line up along the middle of the cell, and the spindle fibers are going to kind of hold them into place. Next is anaphase, A for apart. So the chromosomes are going to be separated into individual chromatids, and they will get pulled apart towards the ends or the poles of the cell. So they move into the opposite direction. So A for apart. Telophase then is going to be when we have the nuclear membrane is going to start to reform around the chromosomes at the poles, and the spindle fibers are going to disappear. At the end of telophase, you have one cell that has two nuclei in it. And what's going to happen, you can see this right here, is this cleavage, is that the cell is going to cleave or split into two new cells right around this cleavage line, and then that's going to have this nucleus, and then this cell will have this nucleus, and because each chromatid is exactly the same, they would be identical. So cytokinesis is when that cleavage furrow forms and the cell divides into two identical cells, each one with a nucleus, each one with an exact copy of the chromosome, exact copy if everything went well. Here are some slides of mitosis and the cell cycle. So this is going to be interphase. I always think interphase looks a little bit like a fried egg because it's got these big dark kind of spots in it, and you shouldn't really be able to see any lines. This right here is prophase. You can start to see the lines forming. These are the chromosomes. Here is metaphase because the chromosomes are lined up along the middle. Here's anaphase because they're pulling apart from each other. And then here's telophase where you have like this one cell with two nuclei in it. Your book talks about cyclins and the importance of them. These are going to be groups of proteins that are found in the cell that regulate the timing of the cell cycle. So basically it's going to tell the cell when to go from G1 to S, when to go from S to G2, when to go from G2 to M, and it's going to tell it how fast to go through. We put cyclins into two categories, internal regulators and external regulators. Internal regulators are going to tell the cell when to proceed from one stage to the next, so G1 to S, S to G2, G2 to M. And then external regulators are going to tell the cell to either speed up this process or slow it down. Cancer is a disease of the cell cycle. The cancer cells do not respond to those internal regulators or those signals that regulate the growth of cells. So it actually is a problem on two accounts. We have cells that are going to move through the cell cycle without the proper checkpoints, so the DNA might not get copied correctly. So we have potentially genetically mutated cells. The second problem is that the external regulators don't work, and so the cell starts going through this cell cycle very, very quickly, forming more and more and more and more cells, which ends up becoming a tumor. A tumor is a clump of cancerous cells or cells that have been damaged. So cancer is a problem of both types of cyclins, both type of regulators. One of the genes that causes a significant number of cancers is called the p53 gene. This gene normally pauses the cell cycle until all of the chromosomes have copied themselves. If the chromosomes can't finish that process, they become damaged. And so if the p53 gene is not functioning, then the cell cycle will not pause. It'll just keep on going through, making more and more genetically damaged cells. p53 gene, or a defect in the p53 gene, causes a ton of different kinds of cancers. Here you can see some tumor masses in the pictures. Normal brain. There's a brain with a big tumor mass in there. This is also a bone tumor mass. Last thing I just want to talk about real quickly that your book does not get into very much, but I think is important to know, is what happens to lead to cellular death. There are two types of cellular death, apoptosis and necrosis. Apoptosis is a programmed cellular death that does not harm the organism. So basically the cell recognizes that it's old and it cannot continue to go through the process that it needs to, and so it's almost like, for lack of a better term, a very peaceful cellular suicide almost. It decides that it's going to start putting into play different things that will lead to the cell basically dying and being reabsorbed by the organism. So the things that happen in apoptosis is chemical signals will be released that will tell the cell to start to shrink. It will cause the nucleus to start to break apart. The DNA will start to break apart, and then it will send a signal to nearby white blood cells called phagocytes, which will basically start eating the cellular debris from the nuclear fragmentation, the DNA that's floating around. And then at the same time, it will release another chemical to surrounding cells to tell those cells to maybe go through the cell cycle at a little bit quicker pace for a time being in order to replace it. So it's a very organized, non-destructive way that the cell will end its life. Necrosis is the opposite. It's an unnatural death of a cell or tissue. So if you've ever heard of someone that's had necrosis or necrotic tissue, here's an example of a necrotic wound. Basically what happens is the exact opposite. Instead of the cell like shrinking and having this controlled death, what happens is the cell basically starts to swell. It starts to get bigger and bigger and bigger, and it explodes, leaving a bunch of cellular debris. And that debris isn't immediately recognized by those white blood cells, so it ends up floating around and releasing toxic chemicals, which will then cause nearby cells to also get bigger and bigger and explode. And basically this cascade effect, or this, think about dominoes falling, is that one cellular death in this way is going to cause all sorts of cellular death in the general area, and it happens very quickly, too fast for your white blood cells to kind of get in there and clean it up at the rate that they want to. And so oftentimes you end up with a wound People who have necrotic wounds have to be treated very, very aggressively and very, very quickly, because it can very easily lead to the removal of a limb. Like, that's how fast the tissue can get damaged, from the skin all the way down through your muscles into your bones. So that's kind of the cell life cycle.